Well, good afternoon, everyone. Welcome to a special Wednesday afternoon lecture that carries the name of DeWitt Stetten. So this is the annual DeWitt Stetten Jr. Lecture. For those of you who wonder who DeWitt Stetten was, he was the third director of the National Institute of General Medical Sciences. And to mark its 20th anniversary in 1982, this lecture was named in his honor. Stetten was someone who had a strong commitment, as you might guess from NIGMS, to basic science, and certainly especially research in genetics, cellular and molecular biology, and chemistry, all of which I think feature quite nicely in the presentation you're about to hear from today's Stetten lecturer, Christina Dawn Smolke of the Stanford University. She received her undergraduate degree from USC, went on to receive a PhD at the University of California at Berkeley in chemical engineering, stayed on for a bit to do additional postdoctoral training at Berkeley, and landed at Caltech as assistant professor in the Department of Chemical Engineering. Did wonderful work there for a few years and then moved over to Stanford, where she was initially assistant professor, then associate, and is now full professor in the Department of Bioengineering and by courtesy of Chemical Engineering. We're proud of her because she has been somebody who's done great things for biomedical research, and we've been able to crow about it. I have blogged about her work on occasion, and I'm sure you all watch everything I write in my blog, so you already knew that. She has also been a recipient of the NIH Director's Pioneer Award, which is a particularly prestigious award for people who have bold ideas and aim to pursue them boldly. And that was on synthetic biology platforms for natural product discovery and biosynthesis. You might have noticed some of her work back in 2015 when it was her lab that basically figured out how to put 10 different genes from 5 different organisms into yeast and convinced the yeast to synthesize hydrocodone, an opioid, basically from carbohydrates, which got people excited and a little alarmed. But this was not Breaking Bad. There was actually a limited amount of the substance being made, but it was quite a revealing moment to see just what could be done with creative engineering in terms of harnessing the yeast organism to begin to look at becoming a drug manufacturing system. And Dr. Smolke has gone on to be co-founder and CEO of a company, Anthea, which is leveraging that kind of advance in synthetic biology. But today, she's going to be talking to us about another area of her research, which focuses on RNA as a switch, as a sensor, a controller, and how to generate scalable platforms of that sort. I think you're in for a pretty exciting romp through some very creative engineering using RNA as the molecule and reflecting the work that she's now doing at Stanford and as a Chan Zuckerberg BioHub investigator. So, would you please join me in welcoming Dr. Christina Smolke, who's going to talk to us about scalable platforms for generating RNA sensors and controllers. Please.
Thank you for that really kind introduction, and it's really a pleasure to be here and an honor to be giving this lecture to you all today. I'm going to focus the talk today on giving an overview of area of research in my laboratory that has been funded very nicely through NIGMS, through R01 awards. And so it'll be more recent work. Some of it was recently published. Some of it is actually unpublished, but I hope to be published in the next few months. And I'm looking forward to the questions and discussions that will follow. And so this is an area of work in my laboratory where we've been very interested in thinking about how we can develop essentially gene expression control systems that have inherent properties of programmability that we will be able to take into different types of cellular systems and be able to use them across different networks and towards different applications. So I want to start, and the talk today is going to focus more on some of the new kind of foundational engineering approaches that we've developed that allow us to try to scale our approaches for designing tailored genetic switches that are made out of RNA But before I jump into that work, I want to give maybe a little bit of a motivation of why we're interested in being able to develop these types of tools for biological systems. And so we're going to be talking about a broader category of RNA switches today, which is a class of genetic switches. Specifically, we'll focus on RNA switches that utilize a ribozyme-based cleavage mechanism, and I'll go into what that exactly means over the next couple of slides. But taking a step back here, I really just wanted to highlight a few examples that have been published in the literature that have utilized these classes of genetic switches to be able to program dynamic and conditional control of gene expression in biological systems. And so a number of years ago, there was some work in the field that essentially used these types of switches to be able to control proliferation and activation of T cells within model systems. And this is work that has actually now been licensed and is being commercialized in a number of early stage cell therapy companies. But again, towards the general purpose of being able to basically tune and conditionally control the activation of immune cells in a person's body. Other examples have focused on being able to control viral infectivity. So being able to basically control the infectious state of a virus and its replication within cells, which has a lot of applications for gene therapy, as well as control over the cell cycle, has been sort of an additional application, and that has a number of interesting implications, as well as if we can basically pause and control how a cell walks through its different stages of cell cycle. And then finally, with all the interest in sort of genome engineering, genome editing that has been recently in the field with CRISPR-Cas, there have been a number of examples of people leveraging these types of RNA switches to be able to achieve conditional control over genome editing, which is also important if you're thinking about ultimately therapeutic applications for genome editing as well, being able to precisely control that and turn that off and on. Okay, so with those types of broader applications in mind, particularly in the health space, I want to now take a step back and focus the talk today on how do we actually engineer these genetic switch systems so that they can give us the type of performance we want when we move them into different cell systems. And so I'm going to first use this slide to walk through the mechanism, basically, of how this RNA switch and ribozyme switch works. And so essentially showing you here basically the architecture of a transcript, a messenger RNA, as we would have it in a eukaryotic cell. And essentially what we do is we place the ribozyme, which is this sort of 2D structure that you see here, in the 3' untranslated region of our gene of interest. So this can be any actually endogenous gene or transgene that we then insert this ribozyme switch into. And the basic idea is that in sort of a baseline case, in the absence of any conditional inducer, that ribozyme, this is going to be a self-cleaving structure of RNA, so the ribozyme will actually self-cleave. And when it self-cleaves, it opens up the transcript, which allows it to go through very rapid degradation. And then essentially, we're in what we would call a gene expression off state. So because of rapid degradation, protein expression is reduced, and effectively gene expression is off. Now, this ribozyme switch has a sensor domain, which can actually bind a molecular ligand within the cell. When that molecular ligand binds to the ribozyme sequence, basically what happens is that disrupts the ability of that ribozyme to cleave. The transcript is effectively stabilized, so you get more copies of it at its steady state level inside the cell, and thus you get higher levels of gene expression. So that's effectively how this genetic switch is going to work in this sort of off state, right, and then being able to turn on expression when the ligand is present or is present at elevated concentrations inside the cell. Now, if we think about sort of the architecture of this RNA switch, we've done a lot of work in terms of thinking about how we take more complex genetic devices and separate them into their underlying component functions. And so in this case, what we essentially have is 2 component functions. We have the ribozyme, as shown here, and then we have the aptamer, which is going to be our sensor domain.
The aptamer is going to be the segment of the RNA that binds the molecular target. And then we basically will physically couple the ribozyme and the aptamer, as you see here on the right-hand part of the slide. So essentially what we do is for the ribozyme to cleave under physiological conditions, there are loop-loop interactions shown here by these dotted lines, which have to form in order for the ribozyme to go into a cleavage-competent state. We actually replace one of those loops with the aptamer domain, as shown here, and we modify the sequence of the corresponding loop so that it still maintains those loop-loop interactions so that it can form a cleavage-competent state inside the cell. But the idea then is that when that molecular effector is present and binds the aptamer, it disrupts the ability of those loop-loop interactions to form, thereby disrupting the cleavage activity of the ribozyme. Okay? So that's effectively then the mechanism of how the ribozyme switch works, and also the architecture of how we think about these two different components coming together, the ribozyme self-cleavage activity, and then the aptamer ligand binding activity. And so today I'm breaking my talk into two different stories, and they're basically stories that are focused on different aspects of how we can sort of develop higher throughput methods to tackle the design process. And the first part is going to go over basically some new approaches that we've developed that are based upon next generation sequencing technologies that allow us to very rapidly engineer and optimize these switches for activity within different mammalian cell types. And then the second story will be an evolutionary platform story that discusses a new system and workflow for evolution that we've created that allows us to scale the generation of new biosensors, right? Those sensor domains, which has been quite limiting to the field. And we'll talk about that in the second part of the talk. So let's go into the first part of the talk. So if we think about how do we design these ribozyme switches, right? What the design challenge is. Essentially, again, the way that this works is that when we basically graft, if we start with a known aptamer, so a known sequence of RNA that binds a ligand of interest to us, if we graft that onto one of the loops of this hammerhead ribozyme, the design question then becomes, what is the optimal sequence for that second loop such that it will basically reenact or maintain those loop-loop interactions, right, between now the aptamer bulge and this redesigned second loop. So what is the optimal sequence, right? Because it's going to be different than the wild-type sequence because you've changed the loop on the other side. And so essentially, if you think about this from a rational design perspective, the softwares that we have available for us are quite good at predicting sort of secondary structure folding of RNAs, but they're not very good at being able to predict these types of loop-loop interactions. So it's not very tenable, basically, to be able to just sort of specify what that loop sequence should be for a given aptamer. So we instead will default to library screening strategies, where we will take this loop region and basically just completely randomize it, and then try to screen that library within a mammalian cell system to identify sequences that give us strong cleavage activity and also the ability to respond to that ligand in a conditional manner. And essentially then there's two criteria here. What are the sequences, right, that allow interaction with the loop 1 aptamer to facilitate cleavage under physiological conditions? And also, what are the sequences that meet the condition of allowing ligand binding to disrupt that cleavage activity? And so we looked at and developed two parallel seq methods to be able to do this screening under a single experiment in a massively parallel manner. And I'll walk through both today, but the primary focus is going to be the RNA-seq method because that actually winds up being the more streamlined and efficient method in terms of resources and researcher effort. But we looked at both RNA-seq and FACS-Seq, and basically RNA-seq is going to read out activity levels based upon steady-state levels of the RNA target within the cell, whereas FACS-Seq is going to read out activity levels of the switch based upon protein levels within the cell. But what you'll see is that both effectively work in terms of a screening strategy. Okay, so first we'll focus on RNA-seq, and this is essentially the workflow that was developed. And so essentially, although there are a number of steps here, it actually winds up being a fairly streamlined and rapid workflow, because once we designed that library, was again essentially grafting the aptamer onto one of the loops of the hammerhead ribozyme and randomizing the second loop. We just clone this into a plasmid library, we transfect it into our cell type of interest, and we do a transient assay here, basically. So we're not doing stable transfections or transductions. We do a transient plasmid transfection, and we go through a workflow where in one sample set, we just extract the plasmid DNA because we're going to normalize that to our counts. In the second sample set, we'll extract the RNA from this transient transfection. We'll do a reverse transcription off of a targeted cDNA. We'll do barcoding and PCR prep for the NGS prep, and then essentially do Illumina sequencing, where we're able to, through Illumina sequencing, basically count the occurrence of each individual ribozyme switch at the RNA level, and then also we normalize it to the DNA level, which allows us to normalize for any variation in plasmid transfection efficiency and occurrence on the DNA. And so that's the workflow that we develop. It takes us just a couple days to actually perform the experiment, but because we're using a seq method, that NGS, next generation sequencing approach, can basically read out tens to hundreds of thousands of different sequences, right, within the library population. And through the seq approach, we're just counting the occurrence for each individual sequence as a way to read out relative levels. So we did this first and developed the method and validated it using a model aptamer. This model aptamer is the theophylline aptamer. It basically is a sequence of RNA, as shown here, that binds to this purine alkaloid that you see here. Okay? And so we then created a library where this was grafted onto each loop. We randomized the second loop in an N4 sequence. And then basically we performed the workflow that I just walked you through. Transformed that library into our cells of choice, did the assay in the presence and absence of theophylline, and then did the seq method to count the occurrences for each sequence in the library. This is what the data look like. And so essentially what we're plotting here then is the normalized counts for RNA to DNA ratio in the presence of theophylline, and then on the other axis in the absence of theophylline. This dotted line is really just the one-to-one ratio. That would be where you would expect sequences to fall that don't respond to theophylline. Okay, and that's those little crosses that you see, those are basically our controls, our ribozyme controls that we don't expect to respond to theophylline, and you see that they fall very nicely across that line, and they're tailored to have different levels of cleavage activity, which is why they fall across that line in different levels. And then what you see is that there's sort of every data point here, every circle is basically, or every dot is basically a different member of this library, a different ribozyme sequence. So again, in a single assay, we're able to read out the entire activity profile of every member of this library. And basically what you see is that there is a distribution of activities, right? And the things that you would expect to be sort of the strongest switches or the strongest responders are in this quadrant here. What that means is that they have low levels of activity in the absence of ligand, and then high levels of activity, right, or they're responding to ligand when you add it. And so you get higher RNA levels, right? So this would be the area of the library that we're very interested in examining and validating. And then what you can see here is just sort of a trend over on the right-hand side, which is looking at sort of the fold change in activity, which would be basically the ratio of the number on these two axes as a function of the basal state of a particular sequence, which is basically its RNA-DNA ratio in the absence of ligand. And as you would expect, the things that are going to perform as the best kind of switches are things that have low basal levels of activity, or basically high cleavage activity in the absence of ligand, right? And so again, that's sort of these sequences here. We can then go in and we have the sequences, right, that correspond to each one of these data points. We can go in and basically validate them individually through a flow cytometry assay, right? So this is just an independent validation that the RNA-seq is basically giving us the correct measures of activity for each individual switch. And this is essentially what the flow cytometry assay looks like. You take each individual sequence, you use it in the same expression platform, express it in your cell type, and then read out individually via the expression of mCherry ratio to BFP what the level of activity is. Where again, in this case, what we're doing is using that switch or the ribozyme switch to regulate mCherry, and then BFP as a normalizing control to normalize for noise and gene expression across the cell population. And again, this is the type of data that you would see, where this is a histogram for an individual switch in the absence of ligand, and then the presence of ligand here so that you can see that it's responding to ligand. So we do this basically to validate the individual switches, and this is essentially data from doing that for the theophylline library, right? And so we just picked out particular members of the library, tried to get them to span a range, not just the best activity switches, which would be sort of on this side of the library, but you can see basically, and this shows basically the correlation between the flow cytometry validation data and the FACS-Seq data. And you can see that there's very good correlation between the two measurements, which validates basically the RNA-seq approach of doing this type of assay readout. And then what you can see basically is in the flow cytometry data, the activities of the validated switches at absence of ligand, 1 millimolar theophylline, that's what we feed the cells, so the intracellular concentration is quite a bit lower, and then 5 millimolar theophylline just to see if there was any additional response. You could see that most of these are tapped out at 1 millimolar, which is what we would expect. The controls here are sort of the fully active ribozyme control is what you see on the left, and then the inactive ribozyme control is what you see on the right. So that would be sort of bracketing the high and the low activities of the ribozyme switches that we would expect to be able to get. And you can see that we're approaching basically fold changes in activity at the flow cytometry level of about sort of 10-fold here, and getting pretty close to sort of the fully off state of the ribozyme by this type of measurement. Okay, so then what we wanted to look at was how extensible, right, or generalizable this particular workflow was, and in particular, how well it worked for aptamers of different architectures, right, and different structures. So we took 3 additional known aptamers to small molecules in the field, basically the hypoxanthine aptamer, which is also a purine alkaloid. We took a fulinic acid aptamer, which you see here, and then also an aptamer to cyclic di-GMP, which had a very different structure from the other two. So trying to get both diversity in terms of the ligand itself, as well as what the aptamer structure looked like. We applied that workflow to each of these, and basically the RNA-seq data sets that came out of those looked quite similar to what we saw for the model theophylline aptamer, right? Again, in a single experiment, you get data on every single member of the library, as you see across each of these plots. And as you see, they all have very similar sort of trends, which is sort of just a very horizontal kind of trend coming off the x equals y sort of line here, with the best switches, or the switches with the strongest, at least fold change, would be over in this side of the quadrant. And the hypoxanthine and fulinic acid look quite similar. Cyclic di-GMP is a little bit different from the sense that it's closer to that diagonal line, right? You can see that as sort of a difference between these. The reason for that is that the ligand, cyclic di-GMP, actually doesn't get into the cell very well at all, so it's very hard to actually activate the switches. You can get pretty strong ribozymes, but it's hard to disrupt that cleavage because the intracellular ligand concentration is quite low. What we reported on in this study is that fulinic acid actually initially looked quite similar to cyclic di-GMP, and we were able to improve it by actually engineering transporters for fulinic acid into the cells so that they could uptake the ligand and have higher intracellular concentrations to basically activate these switches, and then saw a better response overall of the library. Again, we could validate candidates from each of these libraries, just as we did with the theophylline, and basically demonstrate that the things that came out of the RNA-seq were directly validated and had very good correlation with the flow cytometry measurement, as you see here. And again, what you would see again is that the hypoxanthine system performs pretty well, and quite well. Fulinic acid certainly performs also reasonably well. When you get to the cyclic di-GMP, we basically, again, have issues with being able to activate the system in a very strong way because that ligand does not get into the cells very well and is present at really low intracellular concentrations. And so that's the essential limit with that particular aptamer, not the structure or the workflow method itself. So the next thing we wanted to look at with this particular study was, the RNA-seq is basically giving us a readout of what's happening at the RNA level. And of course, a lot of times what we actually care about in designing these systems is how that transfers to a change in protein expression. And so we wanted to ensure that the things that we would be picking out by measuring at the RNA-seq level, that we weren't missing anything by basically not measuring at the protein level, right? That we weren't leaving kind of good switches on the table by not doing a FACS-Seq approach. And so we analyzed the same libraries with the FACS-Seq approach to just do that comparison and see. Now, one of the reasons that we might not want to directly go to a FACS-Seq, or probably would like to avoid it if we can, is that it's a much more cumbersome workflow in the sense that to do a FACS-Seq, you can't do it via transient transfection. We have to do it through a stable integration. And we do it via a stable integration by leveraging a lentiviral platform. So the same library is basically then cloned into a vector that allows us to do lentiviral packaging as well as transduction, so there's additional steps there. We then have to transduce the library and grow out kind of stable clones from that library, trying to get very close to kind of single integrants into each cell. And then once we go through that process, we can go through our FACS spinning process, which essentially then takes that library that's now stably integrated. You analyze it in a FACS experiment where you're measuring the ratio of mCherry to BFP, and you're binning in these different activity bin ratios, as you see here. You then take each of those activity bins, you recover the cells, you then do PCR and barcoding of that ribozyme region. And again, you reconstruct the histograms by just counting the number of occurrences of that particular sequence within each activity bin. Okay? So it still gives you data on every single sequence in the library, in a single experiment, but it is a much more cumbersome and longer workflow. So when we did this, what we saw first is that the data on the left-hand side is just comparing replicates. So you can see that we get good replicates between repeat experiments. The data is very reproducible, which is what we would like to see. You can now, if we look at what the data looks like in the absence of ligand with the presence of ligand, you might remember with the RNA-seq method, the data came out much more horizontal here, right? Here, it's much noisier, is basically what we observed with the FACS-Seq data. So there's a lot of noise in the data. A lot of things are falling along the line. I'm highlighting for you in yellow the switches that were identified from the RNA-Seq data where they fell in the FACS-Seq data. So you could still pull them out if you were looking for things on this side of the line, but it's definitely not as clear of a signal. So if we look at how it performed with the other libraries that we tested, the story is very similar, right? Basically, you could see for each one of these graphs, the data looks a lot less clean. There's a lot more noise in the distribution. It doesn't come off sort of in the clear horizontal line as it did for the other libraries. But you are able to see, and you would be able to pick out switches, functional switches within the library, as you can see by the circled data points here. What I'm showing you in these little overlap circle plots is basically the overlap in the activity ratios between RNA-seq and FACS-Seq. And so you can see that there is a lot of overlap, but there are some things that FACS-Seq would miss. And so then the final thing that we're able to do, so one of the nice things about doing these types of seq experiments and getting data on every single member of the library in a single experiment is that you can begin to use that data to draw out basically design principles, right, for the sequence function, the sequence activity design landscape, right? So you're not just getting data on a small number of sequences that act, optimally with whatever your screen is. You get data on the full library, and then you can analyze that and begin to draw out patterns, right, in things that have strong activity versus low activity, and what this sort of sequence relationships are like here. And so what I'm showing you here is analysis on some of those large data sets that we did. For each of the libraries, you could see theophylline, xanthine, flinic acid, and then actually we did cyclic di-GMP for 2 different known aptamers. And you can see basically the consensus sequences in the loops that were generated based upon the loops that gave the highest ribozyme cleavage activity for that given ribozyme. And you can see that there are certainly consensus sequences that pop up. And generally, they are different across the different aptamers, which is what we would expect because those aptamers are going to have different sequences themselves. And you can also see then for each one, this is data that basically shows, again, the population average RNA DNA sort of activity level for different consensus sequences that are shown below. So the one on the leftmost for theophylline would be no filtering of the library, the full library, and you could see then as you sort of filter more towards the consensus sequence that you see here, you're basically reducing the activity profile of the library, which is what you would expect to get the highest cleavers, right? And so it also allows you to pull out the independent effects of each nucleotides across different regions of the loop and how they contribute to that overall low gene expression profile activity in the absence of ligand. The idea then is that as we and others continue to generate these data sets, right, and begin to be able to generate larger and larger data sets, we can begin to continue to do these types of analyses. And one of the other things we're doing in the lab right now, which I won't talk about today, is actually leverage these data sets with machine learning types of approaches to be able to develop tools that will allow us to actually do forward and rational design of ribozyme switches based upon using programs that will learn what these interactions should be from these large data sets and be able to forward predict them. And we've seen basically actually very interesting results and success with these machine learning models, being able to forward design with aptamers that it has the program hasn't seen before. Okay, so final thing just to say then, again, one of the things that we wanted to do with this is really compare the workflows of an RNA-seq and a FACS-Seq method in mammalian cells. And what came out of this in terms of a conclusion was really validating the RNA-seq approach, where I think initially when we started there were questions about whether reading out RNA levels versus the protein levels was going to be effective and whether we would miss things. There's definitely a big advantage in terms of just the effort, time, and resources. The RNA-seq takes about a week. FACS-Seq takes well over a month. You can use the RNA-seq with off-the-shelf kits, just access to a sequencer. For FACS-Seq, you need to have access to a FACS sorter or external facilities that give you that access. So it's a much more cumbersome process. And in fact, at the end of the day, the data from the RNA-seq actually had less noise and was a better predictor and clearer predictor of the activities of these switches inside cells. So now, so that story was really focusing on, given an aptamer sequence, how do you go about and what are approaches that allow us to quickly and efficiently develop a RNA switch that will work in a mammalian cell based upon that aptamer sequence. The next thing the next story is really going to focus on, okay, what if you don't have an aptamer sequence, but you still want to develop a biosensor that you can use inside of different cells? How do we go about really streamlining and parallelizing the workflows that we use to develop cell-based biosensors to new ligands? And that's going to be the part 2. Okay, now really what the challenge is, right? What is the challenge? The challenge is really in terms of the diversity of sequence space that you have to be able to effectively search for these two different problems, which is why we can't just use the part 1 approach to develop new biosensors. Essentially, the library sizes in the part 1 approach, where you have an existing biosensor and are just trying to optimize a switch, the diversity sizes there, are going to be on the order of maybe 10 to the 4 sequences, right? Because you're randomizing that loop region of the ribozyme. If you have to go forward and actually develop a new biosensor and really generate the new sensor component of that aptamer within the ribozyme platform, you have to search a much larger sequence space. And that sequence space is going to be on the order of 10 to the 14 to 10 to the 15 sequences to really be effective at generating new biosensors. That's a huge shift in sequence diversity, and it also means that you cannot directly use in vivo approaches, right? Because in vivo approaches, you're going to be limited by the transfection efficiency of the cell, and nothing will get you to sort of that diversity. So you have to be able to develop an in vitro method that will still be able to transfer and translate to the environment of the cell, right, in terms of what it's selecting. Okay, so first want to talk a little bit about the challenge here and how really this is kind of a fundamental limitation of the field that I work in and others work in. There's a lot of talk about programming biological systems and how to combine, sensors into these programmable gene circuits. But at the end of the day, if you look at it, we're really limited in the types of sensors and switches that we have to build these circuits, right? And so a lot of times you'll see people reuse many of the same components, but as we want to go into new applications and new systems, it becomes a major limitation. And so if we just look at the aptamer space and basically sort of look at the small molecule inducers that people have used regularly in synthetic RNA switches, this basically breaks it down here. You'll see about half of the literature has used the theophylline aptamer. Another about 20% has used a tetracycline aptamer, and then there's a handful of others that you see present in the literature, right? But it really is a small subset, given that CLEX was developed a number of decades ago, right, and has been applied in terms of just the number of aptamers that are available for us and that are validated to work inside cells in these types of systems. The idea then is that also, once you have an aptamer, as you just saw from the first part of my talk, integrating that aptamer into a switch and optimizing it for performance in a cell is also not trivial, right? So you have this sort of two-step kind of design process that currently takes place. Now, I think if we look at sort of traditional selection for aptamers to small molecule ligands, there are a lot of kind of common challenges, I think, that are present in the process, which make it particularly difficult, again, when you're trying to develop aptamers to small molecules. And this gives an overview of what that process looks like in a traditional sense. So you start with your nucleic acid pool, you start with a diverse pool on the order of 10 to the 14, 10 to the 15 different sequences. For small molecules, though, the challenge is that you oftentimes have to conjugate your small molecule to a solid support so that you can do that partitioning process. That effectively changes the presentation of that small molecule, right? Because that's not how it's going to be presented to the aptamer in solution and in the cell, right? And that can actually lead to problems in terms of what you're actually selecting for. But at the end of the day, then what happens is you incubate these two. That sort of solid support is used then to specifically remove non-binding pieces, right? Retain those that are binding the small molecule. As you see here, you can then elute from the target. You recover the small number of sequences that are binding to your target, which is conjugated to a solid support, and then you use amplification to take it into the next round, right? And you can go through many iterations and cycles of this. Again, I think the big challenge here, at least as I've seen it and tried to put it into place, is that this solid phase immobilization step creates a lot of challenges. It creates challenges in terms of you're removing functional groups that the aptamer might ideally interact with. It also means that that's what you're selecting for is a small molecule conjugated to that matrix and not in solution where it might typically be presented. The other thing is that this overall approach then becomes fairly labor intensive and not scalable when you're actually doing this in the laboratory, meaning as I've seen it be enacted for small molecules, a single researcher would do a selection to one molecule at a time, maybe two, but it's very hard to do, say, 30 to 40 at a time, right, and scale it that way because it's not a solution-based process. So what we did is we really looked at this and tried to change the evolutionary and selection platform to one that would be solution-based. And once it's solution-based, that also allows you to then automate it on a liquid handling platform so that you can run many selections in parallel without researcher interference. And so these are the steps that I'm going to go through with you today in terms of this overall process that we developed, which we call DRIVER, for de novo rapid in vitro evolution of RNA biosensors. It starts, as you'll see, with the inputs, which are essentially going to be the RNA library and your ligands of choice. It goes into this in vitro evolution sort of cycle, which we switch to be a solution-based method, and so that we can automate it on a liquid handling robot. The activity and the enrichment process is basically read out through a Seq assay, which I'll describe, a Cleave-Seq assay. And then finally, at the end of it, the idea is that that allows us to identify novel RNA biosensors and to do this in a parallelizable workflow. Okay, so let's take the steps one at a time. The first is the library design and the small molecule targets. So basically just piggybacking off of the first part of the talk, we're going to use the Ribozyme switch platform that I described. But in addition to randomizing that loop that the aptamer will interact with, we're also going to randomize the aptamer loop, right? For the purposes of what I'm presenting for you today, what we did is we just randomized the aptamer loop completely in either an N30 or N60 format. You could develop many, many different types of library designs beyond what I'm showing you here. This is sort of the most basic version. And we are testing other library designs as well, right? But this, we decided to start with sort of the most conservative version possible. In terms of the small molecule targets that we started with ones that we were interested in, which were basically plant natural products and intermediates, plant growth hormones, central metabolites, and different types of small molecule therapeutics. Okay, so that's the inputs. Now we're going to step through sort of the driver selection platform as well as the NGS assay. And so with the driver selection platform, what happens is you take that input, you basically do a transcription in the presence of ligand. You're going to have each sequence will either be in an uncleaved or cleaved form, and then you're going to use a process basically, in this case, for the positive part of the selection, to select and amplify uncleaved molecules. That will then create your next library, which goes into transcription in the absence of ligand, right? And again, here you're going to have the library members being either in an uncleaved or uncleaved state, but here you're going to select for cleavers, right? So you have basically a round of positive selection, negative selection going into the next round. And you can modify the conditions in terms of the time you allow for transcription, magnesium concentrations, et cetera, which allows you to modify the stringency of this process. But again, the core idea here is all of it is selection-based. What you'll notice is that we're shifting, right, from developing a selection that's based upon binding, right, to one that is based upon cleavage, right? And basically whether or not this ribozyme switch sequence is cleaving or not cleaving under these different conditions and at what rate, which is effectively what we want in these biosensor platforms that we're going to use. The NGS assay is basically going to be used in a very similar way to the RNA-Seq assay that I just described, except it's not done inside cells, right? So instead of having to transfect your library into a cell, you essentially take that same transcription process I just described, and at any point, you can just use a seq method to read out the number of times a particular sequence occurs in a cleaved and uncleaved state. And that you can use to basically back out cleavage rates for each sequence, and you can monitor them as you go through the course of the evolutionary rounds of cycles, what's happening with enrichment in your library. Now, all of this, both the selection process and the Cleave-Seq process, are basically driven and enabled by a novel regeneration method that I'm going to just walk through in this particular slide. And really just focus then, the novel regeneration method is sort of focused on this part of the slide right here. This just sort of shows that it goes into both. And really, what is the core challenge? The core challenge is when you're doing a reaction like this and you have uncleaved RNA molecules and cleaved RNA molecules, it's actually fairly straightforward to recover the uncleaved because it has a unique sequence on its 5' end as you see here. Right? So the uncleaved will not have this portion of the molecule present. So it's fairly straightforward to recover that in a mixed pool. It is actually quite difficult to enrich the cleaved molecule from a library because the 5' end of the cleaved molecule is the same, it is also present in the uncleaved. So you can't just design a primer sequence that will amplify selectively the cleaved molecules away, whereas you can for the uncleaved. So what that means is we developed this sort of unique regeneration process that would handle both. And so basically what it comes down to is if you look at the primer anneal step, we created this sort of unique primer that you see here. It's fairly complex. But the basic point of this primer is that it will bind to both the cleaved and uncleaved. You do a reverse transcription. Now, for the uncleaved molecule, or for the cleaved molecule, what happens is the reverse transcription will stop here at this point. That primer still has this closed end, and you'll be able to basically create a circular ligated product, as you see here, only for the cleaved population. In the uncleaved, you're going to extend further, and so you won't get that closed end on the primer, and you won't create a circular closed RNA product, right? And so basically what happens is once you have the circular closed RNA product, we insert some U's into the primer. We use a user digestion, which cleaves at the U sites. And so basically after the cleavage, you basically recover this cleaved molecule with this new barcode shown in green put on it, which we can then use to basically PCR and develop and specifically recover, right, the cleaved molecules now with the green barcode. And with the uncleaved, It's not being switched over to the green barcode, right? Because it wasn't able to go through that circular ligation, but we can still uncover it, and it has a unique barcode that we use here. That allows us then to both use those unique barcodes to count the occurrence of a sequence in a cleaved and uncleaved state for each reaction, as well as recover specifically the cleaved or uncleaved molecules by those unique barcodes and take them into the next round of selection. So that was really the key here for DRIVER to be able to work and to work efficiently. Now, what we did then is we took that process that I just showed you and automated it on a liquid handling robot. So all of these steps of transcription, PCR, digestion, et cetera, they're basically all reactions that occur in solution at particular temperatures, pipetting solutions, right, to and from different wells. And so we could just automate this in 96-well plates. And basically what we now do is we have a round of positive and negative selection, can be automated in about a 3-hour time block on the robot. And we can run about 40 hours of selection per week doing that, basically. And the idea, though, is that because this is run on a 96-well plate, you could have many selections running in parallel, right? Because it's all just sort of being solutions pipetted around. We could look first, this is just some data showing you how enrichment occurs over time in a library. And so basically what I'm showing you here is the fraction cleaved as read out by this process, starting at round 0. And we took a library and basically just went up to actually 150 rounds using this method. That's actually quite a lot. We don't have to do that many, but we just wanted to kind of follow what would happen. And you can basically see that, the pool is enriched in cleavers as we go through the selection. And most of the enrichment, though, occurs in the early rounds of selection. And after that, it fairly much levels off. But so it demonstrates that we are getting enrichment from fast cleavers through this process. Now, the other thing we can do, again, is that as we go through these rounds of selections, we can stop and basically use that Cleave-Seq NGS assay. And this is an example of data from a particular round of selection where, again, what we do is at any time point of enrichment, we basically do a Cleave-Seq that will give us for every, well, not every member of the library because there's too many actually present, but for a represented member of the library, the fraction cleaved in the absence of ligand and the fraction cleaved in the presence of ligand. And basically what we're looking for is sequences that occur off the diagonal, right, in terms of ones that are going to act as switches, showing that there's a difference between these two states. We can take those individual sequences, as you see here in red, and then begin to probe them further. So does this actually work to identify novel biosensors? The answer is yes. We've done our first rounds of selection and application of this. What I'm showing you here is a heat map where on the axis here, you just see different ligands. So there's plant hormones like transietin, plant natural products like theophylline, reticulin, noscapine, and then some small molecule therapeutics, acyclovir, and other small molecule therapeutics that you see here. This is just showing along this axis, each number is basically a different switch sequence, okay? And then they're grouped. So the ones in this group are basically ones that we identified that respond to acyclovir. In this group, respond to noscapine, reticulin, theophylline, transietin, right? So you can see basically that from this, we are getting sensors that respond to these ligands, right? And they do this specifically. We can also use a separate method, specifically surface plasmon resonance, to actually measure the KDs and affinities of the sensors that are coming out of this selection platform. And so what you see here is basically data for 5 different sensors to different ligands, acyclovir, reticulin, noscapine, theophylline, transietin, color-coded, and their binding curves from an SPR assay, as you see here. So what you see, and then over here on this side is basically a graph of the KD as measured by the SPR with the Cleave-Seq, which is basically the EC50 that we measure out through the Cleave-Seq method, and you can see that there's fairly good correlation between different switches. But basically what this demonstrates is that we have validated individual biosensor sequences that do bind specifically to the ligands that we've done these selections for. And these, again, were all done in parallel in a single enrichment experiment. So the next question we wanted to ask is what comes out of DRIVER, does it actually perform inside of cells, right? Can we take DRIVER-selected biosensors and use them directly as genetic switches inside of different cells? And that, again, allows us to go back and tune the conditions of DRIVER to make sure that what we are selecting for can translate directly into in vivo systems. So the first thing that we did was we took the switches basically to some switches to theophylline, acyclovir, noscapine, and transiatin. Didn't make any other modifications to them, cloned them into our plasmid systems where we would use them to regulate, in this case, GFP, normalizing to M-cherry, and just looked at what the activity was. And again, the data on the switch, or not, the control on the left-hand side of each of these is basically the active ribozyme control, no sensor tag to it, so you don't expect it to respond to theophylline. But basically what you could see is that in each of these cases, they do act and exhibit activity inside the cell, meaning that they reduce gene expression in the absence of ligand, and when ligand is present, it increases, right? You'll notice that there's a lot of leakiness in these systems, right? They're still sort of fairly far away from the active ribozyme control. They don't necessarily turn fully on, but at least it's sort of the first initial data demonstrating that they are active inside of cells. We can then, and are going back to modify the conditions of the selection to get them to perform better, to have lower leakiness in the absence of ligand, right? So more stringent off states. But this was an important validation. And we validated in a few other examples. So that was just when we feed the ligands exogenously to the cells, right? Next, we looked at what if we have an application where a cell is actually making a particular metabolite. In this case, we had cells engineered to make reticulin. We had reticulin sensors, and so we wanted to look at could these sensors actually respond to metabolites that the cells are making endogenously versus being fed to the cells. And here, in this case, what you could see is that our reticulin sensors do respond to cells that are making reticulin very specifically and are able to detect increased levels of reticulin produced from our engineered yeast cells. This is sort of the process data, and then the raw flow cytometry histograms shown here. And then as a final example, these two examples were demonstrating activity in yeast. We also wanted to look at whether they would perform in a different cell type, in this case, a mammalian cell type. So again, we took a direct driver-selected biosensors, in this case to acyclovir, and we cloned them into expression constructs that we could characterize in HEK cells. We transformed them into HEK. We basically grew them in the absence and presence of acyclovir just to see whether there was activity. And again, very similar as to in yeast, we do see that the ribozymes are active and are responsive to the ligands, the ones that are being selected directly from driver. In this different cell type, in a mammalian cell type. So definitely, there's still work to be done in terms of modifying the conditions of driver so that we can more effectively basically generate switches that have tighter off states, right, and higher on states. But it's an important validation to be able to take something directly from an in vitro selection platform directly into in vivo. The other thing we can do is that now that we have these biosensors, these new biosensors that didn't previously exist, we can apply the methods of the first part of my talk directly to them to just very quickly iterate and optimize on the activity of the switches, right? So you can use kind of both in parallel once you have the biosensors from driver. All right, and so I'll end there. This is actually a fairly complicated slide, so I won't spend a huge amount of time on it, but really what it's meant to show is sort of, right, the conventional workflow that we have with CLEX, right, traditional CLEX to small molecule aptamers and how we would generate biosensors from them to the driver workflow. And I think, right, the major kind of improvements here are really the solution-based selection, selecting directly within the biosensor platform so that we can take it and move it directly into our cell type of interest as opposed to having to do additional steps once you have an aptamer of then developing the switch. So really streamlining, multiplexing this entire process. And that sort of summarizes my talk for today. I will thank you for your time and attention. And also I want to thank the researchers who really drove the projects that I shared with you today. Joyce Yang was a graduate student in my laboratory who led the first project that I spoke with you about, the RNA-Seq and FACS-Seq work in the mammalian cell workflow. And then Brent Townsend and Joy also led the driver work that I shared in the second part of my talk. Thank you for your time and attention, and happy to take questions.
So thanks for that elegant romp through switch engineering. We have time for questions. Please do use the microphones in the aisles so people watching on video can hear, and we can start right here.
That was really impressive, the solution-based selection and the advantages that that brings. So you were talking about aptamers targeting small molecules. What are the challenges for using aptamers targeting specific protein surfaces within the context of the hammerhead ribozyme?
Yeah, very good question. So the question is, my talk today, both the first and second part, really focused on aptamers to small molecule ligands, and so what are the challenges and differences if you want to actually use that platform to respond to protein targets. So first, we and others have demonstrated that you can use that platform to respond to protein targets. You're just simply putting in protein aptamers versus small molecule aptamers. And if anything, once you have the aptamer to the protein, it is definitely more likely that binding of the protein to that aptamer is going to disrupt cleavage because it's just a bigger ligand, right? So that part actually works a little bit more robustly than it does for small molecules, where you do have that screening approach and checking of how the small molecule is actually binding to that aptamer. So the platform is amenable to that. In terms of the selection platform, you can certainly use driver with protein targets, right? You would just replace the ligand solution of small molecules with your protein target of interest. I guess I would say a few things there. The challenge will just be getting the protein target in purified form, right? You don't have to use purified form. You can certainly use a lysate. I mean, we do small molecule mixtures basically when we use driver. So in many cases, we're not selecting to a single target. In our evolution, we actually put a mixture of targets in there and then do the evolution, at least for certain rounds. So you can use lysates. The other challenge, though, would be getting the protein in pure form if that's what you want to do. And then beyond that, I think with protein selections, of course, it's just a larger binding surface. So, for some applications, you actually want it to bind to specific epitopes on the protein or certain areas. And I think if that's the case, then there are strategies that people in the field have used. Sometimes people will actually do selections to like smaller peptides or smaller portions of the protein that really focus on the kind of area of the protein that you want to bind. The other things that people have done are like for example, there have been some cases where people want to use an aptamer to detect post-translational modifications on a protein. So again, they will do a positive and then a counter selection against the different forms of the protein. But depending on what you're ultimately going for, I think that's going to be the challenge, right? In terms of, and that's across not just driver, it's across any protein-based selection, right? In terms of how you actually do that.
Can you say a little bit more about the evolution approach that you just mentioned? Because when we started out early in the talk about a challenge where you might need 10 to the 14th to 10 to the 15th different combinations in order to really check out the space that you wanted to assess, obviously that's not practical. But if you picked some subset of that and basically looked for weak signals that then could be optimized by further steps of evolution, is that a way that has been useful in this circumstance to try to get from an overly complicated situation to one that still gives you a good answer?
Yeah, it's a good question. And I guess what I would say is, in my read of the literature, it's not conclusive how well that approach works for aptamer development. There's certainly literature and papers that describe both, and this is why I said you can design your libraries in different ways, but there are papers that certainly describe starting with constrained libraries, so really doing a tailored library design so you're really constraining and getting certain secondary structures in there and searching a much smaller space. There's been some examples of success with that, but it's not something that the field has sort of moved to universally, right? There's also been examples where people have started with a parent aptamer that binds a particular ligand and then actually tried to evolve from that parent library, almost in a more directed evolution type approach, to bind to a similar ligand, but maybe that has slightly different set of functional groups. So those have also been described and reported as well. I think what I find a little bit challenging, and actually what we would like to explore with driver a bit, right, is that I'm not aware of any papers that really do a very clear, thorough kind of side-by-side comparison, right, of what happens if you start with this library versus this library versus this library for a set of targets and go through kind of a very similar evolutionary round, which one actually does work better and what are the pros and cons, right? And I think because it's been, in large part, doing these types of selections has taken a lot of energy and resources, and so it's been hard to do them in parallel. One of the things we're interested in with driver is that now that you've reduced kind of the person FTE time and can do a lot of this on a robot, right, and just set up the wells, you can just start to do things like different library designs and take it through the same evolution and see what happens, right, or see what happens if you repeat it. Do you get the same answers? I mean, these types of things. So we're very interested in using it for that as well.
That'd be good. Over here.
Hey, thank you for the wonderful talk, first of all. This is a bit of a tangent, but I like to sort of think about the idealist bigger picture. If I were to say identify a given sensor using driver and want to take it into actual animal use to see if it worked against a given condition like an orthotopic xenograft, right? What are the challenges to the actual use in vivo, like not just cell cultures, but in the actual animal models? Primarily because if you can see this being like taken to the theoretical maximum, you can actually couple the sensor actuator capabilities to like specifically target therapies right when you need them, where you need them.
Yeah.
So
that's, I mean, and that is sort of a bigger picture application of why people are interested in genetic switches and sensors, right? For sure. So your question is, what are the challenges in taking it from what I just showed you to a particular application, right?
Yeah, you showed that the stringent, you didn't really have stringent off capabilities right now.
Not in the ones that I showed you. Although I did show you in the first part of the talk, you can certainly get to stringent offs by just doing more kind of iteration on the design. So, I mean, what the challenges are going to be are basically optimizing the performance of the sensor for your particular application and for your particular cell type, right? And so it's something I think that, what has to happen, and we've worked, we have some collaborations with people who are more clinically minded and, are trying to take these things into sort of early stage cell models, right, to start to move it along that way. But it's my feeling is you have to sort of get early data in the cell types that you're interested in or in the models with what you're regulating to kind of see, where you are with some of that early data, and then you could take that and basically iterate, okay, we need to get we have the biosensor, now we need to sort of reduce that stringency, right? And then kind of do another level of iteration to get the stringency down. In my experience, getting the stringency down is not that difficult, right? But there are these, basically, it's also going to be affected, as you saw a little bit, by what the ligand is, what effector you want to use, right? Is it something the cell is making? Is it a drug that you're putting into the patient, but then what's the clearance rate? What can the concentration really be inside the person? I mean, all these different things basically have to be mapped up when you're in these clinical settings of both what's the stringency, but also how much do you need to turn it on? What are the concentrations of that effector going to realistically be in your different cell types? And so how do you kind of map those to get you into the performance range?
Thank you.
Yeah.
Well, everyone, you're cordially invited to continue the conversation with our speaker in the medical library with coffee and cookies and all that. But first, let's please thank our speaker again for a wonderful presentation.
Thank you.